Plastic pollution is one of the most quickly growing threats to the environment. While it's hard to get a full picture of plastic accumulation when using products on an individual level, the effects are actually immense. Take the Great Pacific Garbage Patch, for example. It's a massive offshore plastic accumulation zone located in the Pacific Ocean at a converging point of winds and currents. It's currently 1.6 million square kilometers large, or twice the size of Texas for reference. This pollution is directly harmful to the environment, the food chain, and ultimately the quality of human life. But there's a lot behind the reason why this plastic pollution is so hard to break down, and that's what we'll examine in today's video. So first, a quick overview of the structure of materials. A polymer is basically a substance made of smaller subunits called monomers. There are natural organic polymers, which are not made by humans and occur naturally in the world, and then there are synthetic organic polymers, which are made by humans and are based on petroleum oil. The degree to which synthetic polymers can be broken down depends on their molecular structure in comparison to these natural ones, which are made from a set of biological macromolecules, including nucleic acid, lipids, carbohydrates, and proteins. Now, for proteins, their monomer is amino acid. It has a central carbon atom, which connects along one axis to a basic amino group and an acidic carboxyl group, and along the other axis, it connects to a hydrogen atom and an R group or side chain. Each amino acid has a unique R group, usually made of carbon and/or hydrogen, that determines its characteristics and function. The polymer of a protein is called a peptide if it has less than 20 amino acids, or a protein if it has more than 20 amino acids. Now, lipids actually have two kinds of monomers, fatty acid and glycerol. Fatty acid has a hydrocarbon chain with at least 4 carbons in its backbone, hydrogen side chains, and one carboxyl group at the end. And glycerol has a 3-carbon chain with one hydroxyl group bonded to each carbon atom. So we'll later see that these two monomer types interact in very crucial ways to form the polymers, which are triglycerides, triacylglycerols, and triacylglycerides, the majority of which are triglycerides. Now, moving on to carbohydrates. Their monomer is a monosaccharide. It's a carbon chain with at least 3 carbon atoms, where one of them is double bonded to an oxygen and the rest are connected to a hydroxyl group or a hydrogen side chain. Their polymer is a disaccharide if there are 2 of them, or a polysaccharide if there are more than 2. And finally, we have the 4th biomolecule, nucleic acid. While nucleic acid is highly important and present in every type of cell, it doesn't bear as much structural resemblance to the other biomolecules or to plastics, so this video won't focus as much on its properties. So now for some info about plastic structure. An example of the most commonly used plastic today is polyethylene. Its monomer is ethylene, which is 2 carbons double bonded to each other, where 2 hydrogens are coming off of each. Here, the degree of polymerization, or how many monomers are linked to each other, is represented by the variable N. This one monomer can actually link to form several different polymers. One of them is low-density polyethylene, which is a chain of ethylene where N ranges from the thousands to 10,000s. It's used for plastic bags and other materials of soft, waxy plastic. Then there's high-density polyethylene, where N equals 10,000 to 100,000, and it's used for insulation, bottles, and materials with a bit harder plastic. Then, even stronger than HDPE is the ultra-high molecular weight polyethylene, where N equals 71,000 to 214,000. Its strength allows it to be used in the medical field for orthopedic implants. So, as an overarching observation, the structure of plastics generally makes them stronger than natural substances, and this strength is associated with their crystallinity, i.e., how aligned and ordered the regions of their polymer chain are. Crystallinity is affected by things like chain length, chain branching, and interchain bonding. For example, HDPE has a long unbranched hydrocarbon chain, where LDPE has a smaller and more branched chain, which helps to account for why one of them makes soft plastic, while the other makes super hard containers. So now we're ready to start comparing these two types of polymers. Our comparisons will involve 2 different methods of polymer formation, condensation and addition. So for a condensation reaction, that's when 2 monomers bond together, losing a small molecule in the process. Dehydration synthesis is a form of this condensation where the lost molecule happens to be water. Now, this reaction is typically observed in the natural biomolecules. In proteins, dehydration synthesis makes peptide bonds form when the carboxyl group of one amino acid connects to the basic amino group of another. In carbohydrates, it forms a bond between the carbon atom of two monosaccharides, linking them together and contributing to the carbon backbone that we see appear. And then we have the same reaction happen again with lipids. As a reminder, the majority of lipids are triglycerides, which have both the monomer types fatty acid and glycerol. Dehydration synthesis causes the hydroxyl groups of the glycerol to link to the hydrogen in the carboxyl groups of each fatty acid chain. So now for the addition reaction. In this case, each monomer has a double bond between carbons, which gets broken so that a bond can form between this monomer and another one. And this process repeats indefinitely such that the monomers are linked to each other one by one to the nth degree of polymerization. This reaction is observed mostly in plastics. A prime example is polyethylene, and we can examine what happens here in steps. The first step is initiation, where a free radical initiator attacks the carbon double bond of the ethylene monomer, starting an electron exchange with the molecule. This results in a single bond between the initiator and one of the carbons, and a new radical on the other carbon atom, which can then be used in the next step of propagation. This means that the new free radical is used to repeat this process as many times as the chain dictates. The final step is termination, where two free radicals contact each other, form a covalent bond, and then get rid of the free radical on each molecule, making the chain complete. Now, a key distinction to make here is that the condensation polymers usually have the byproduct of water, while addition polymers are formed directly without a byproduct. This is because addition polymers generally don't have oxygen, and so most of their reactions depend on the carbon double bond instead. Whereas in condensation polymers, they do have oxygen, and that's really important to perform reactions like dehydration synthesis, where parts of the small molecule that are lost contain this atom, such as H2O. This is also crucial when we consider how the natural versus synthetic polymers degrade. The main mechanism for degradation is hydrolysis, where a molecule of water is introduced and interrupts a chemical bond, breaking a substance into smaller pieces. Basically, this is the inverse of dehydration synthesis, and due to this being their reaction type, condensation polymers can be broken down by hydrolysis in natural conditions, making them far more biodegradable than addition polymers, which can take years to break down and sometimes have to be exposed to excessive amounts of heat or UV light to do so. And hydrolysis also speaks to why the presence of water is so important to biodegradation. Hydrolysis needs water to initiate the reaction of breaking apart the molecule. So in polluted areas where high oil and low water terms exist, e.g. oil spills in the ocean, the slowing down of biodegradation is compounded. So just for reference, hydrolysis in carbohydrates is facilitated by enzymes like amylase, which is found in the saliva and the pancreas. In proteins, it's facilitated by enzymes like pepsinogen in the stomach and trypsin in the pancreas that, when activated in the gut, break the carbon-nitrogen bond between the central atom and the basic amino group. And for lipids, they're broken by a specific form of hydrolysis called lipolysis, which is when triglycerides break back into those glycerol and 3 fatty acid constituents. This is facilitated by the adenylyl cyclase enzyme in adipose tissues, aka body fat. Now, plastic degradation looks a little bit different from this. There are 4 main ways that plastic degrades. Photodegradation, a combination of air and light that initiates oxidation and hydrolysis. Thermooxidation, an exposure to heat or light that initiates polymer chains forming back into radicals. Hydrolytic, an exposure of water-sensitive polymers to water that initiates hydrolysis. And biodegradation by microorganisms, where enzymes within an organism that feed off of the plastic initiate its decomposition. Because plastic degradation is often compared to carbohydrate degradation, the focus of this section will mainly be comparing carbohydrates to plastics. So now we can get to this idea of methods to measure degradation. And it's important to note that alternative methods exist. The following are simply a few of them that are commonly used. For cellulose degradation, it can be measured by a visual examination of cellulose filter paper when placed on top of soil and observed over weeks. And for plastic degradation, this can be measured via scanning electron microscopy, or SEM for short. This is where you shoot a super focused beam of electrons onto the surface of a sample and measure the reflection with an electron detector. The nature of these electrons' interactions with the surface can indicate surface level changes like holes, cracks, roughness, and microbe attachment that all suggest degradation. And these measurements can then be used to calculate and compare the half-lives of different substances, i.e. the time it takes for half of the substance to have degraded. While some plastics like HDPE have a half-life of 250 years, special bioplastics like polylactic acid and macromolecules like cellulose only take weeks or months, so this is orders of magnitude less. The huge difference between these degradation times, as well as other properties, presents a daunting challenge for environmentalists. But after comparing the structure, formation, and degradation of natural polymers versus plastic, we can begin to explore some solutions. One way to approach the problem relies on biodegradation by organisms and the potential for biotech to create organisms that are incredibly effective at eating away plastic polymers. One example is a species of bacteria that eats polyethylene terephthalate plastic, or PET for short, which is used for plastic water bottles and is nearly impossible for other microorganisms to decompose. It was initially found at a Japanese landfill in 2016, where it had naturally evolved to eat plastic by breaking it down with its enzyme PETase. Fun fact, researchers tried studying this bacteria's atoms by shooting X-ray beams at it, but accidentally made it 20% more effective. When harnessed, this could lead to the first instance of full plastic recycling, which would eliminate the need for us to continuously dig up more oil. But there's also another angle to approach this problem of plastic from. Instead of creating organisms that are good at degrading plastics, some researchers are focusing on creating plastics that are good at being degraded. A huge example is polylactic acid. As we've seen, PLA's half-life is only 30 weeks, compared to the polyethylene types that take hundreds of years to degrade, and the difference is rooted in its composition. Unlike other plastics, whose polymers are derived directly from petroleum, PLA polymers are derived from natural carbohydrates like cornstarch or sugarcane. Its production starts with a substance like milled corn. An acid or enzyme is added to the starch to break it into dextrose monomers, which are then fermented to produce the monomer lactic acid, a main building block of PLA. From here, the plastic can either be made directly through a condensation reaction with the lactic acid monomers, or through ring opening polymerization, which involves lactide, a cyclic dimer or ring-shaped link of two lactic acid monomers. The advantage of ring opening polymerization is that it can be done at moderate temperatures instead of very high temperatures like direct condensation. So, while there's still a long path ahead to mitigating plastic pollution, current research and development shows a lot of potential for someday perfecting the answer.